Hello students, in this video session I wanted to explain structure of male gametophyte or structure of pollen grain. So this one we can also say it is a structure of what? Pollen grain. Students, in our last video I explained you the formation of microspores from the microspore mother cell, and after microspore formation this pollen grain, the microspores turn into pollen grain. So what exactly the structure of pollen grain? Look at here, let me take the structure of pollen grain. Each pollen grain, look at here, surrounded by plasma membrane. This is called plasma membrane. So the outermost membrane of pollen grain we can write plasma membrane. Keep in your mind. And inside here in the cytoplasm of pollen grain, abundant food matter will be stored here. So today we will also discuss the pollen grain. Pollen grains also utilized for what? Edible part. It is a energy, food supplementary. Okay. So in the cytoplasm, food will be stored here. And in the middle of the cytoplasm, haploidal nucleus is present. So I can tell you this one is haploidal nucleus. Okay, nucleus. Fine. Outside of this plasma membrane there is intine one layer is there. There is the membrane we call, there is a one more layer we know as intine. So this layer we call intine. Intine, many times in the examination question have been asked, intine is made up of cellulose pectin. Keep in your mind, this layer is called intine. Students, sometimes in the examination they may ask, arrange the layer from out to in. So this membrane I can write intine. Intine is made up of cellulose pectin. Okay, cellulose pectin. This is important for the MCQ. Fine. Outside of intine there is a one more important layer is there. The layer is called exine. The layer which is outside of this intine, like this layer is known as exine. Exine is like this. Okay student, this is called the exine. Exine is made up of world resistant substance that is called what? Sporopollenin. Keep in your mind. Okay, this outermost layer of pollen grain, is called exine. Exine is made up of sporopollenin. Keep in your mind, this sporopollenin is resistant to chemicals, acids, temperature. Am I clear? So this resistant sporopollenin is secreted by tapetum. Am I clear? Tapetum. So what is the function of this sporopollenin? The sporopollenin give protection. It is the outermost layer of the pollen grain. But exine is not completely cover the pollen grain. Look at the pollen grain structure. It is not completely cover. Some places there are the openings are there where there is exine or where there is sporopollenin is absent. Look at here, these are the gaps, these are the areas where sporopollenin absent. These areas are known as germ pores. Clear? Germ pores. Through these germ pores only pollen tube will emerge. Pollen tube will emerge and come out. You get my point? Next, one more important thing. In monocots, if you take the monocot like rice, maize, wheat, the pollen grain has one germ pore. So we can say such type of pollen grain is called uniporate. Keep in your mind, uniporate means one single germ pore is there on the pollen grain. In case of dicots, if you take a dicot, okay, dicot, 3 germ pores are there. 3 germ pores. In where? Pollen grain. Such type of pollen grain is called. Such type of pollen grain is called triporate. Keep in your mind, triporate. Okay, uniporate and triporate. In the examination, MCQ question, uniporate, biporate, triporate they will ask. So monocots are uniporate, dicots are triporates. So here this is the structure of pollen grain. But you need to understand something here. This is the haploid. Okay, this is the pollen grain is haploid. Haploid nucleus is a cytoplasm. Abundant food material also present in the cytoplasm. Exine is there, intine also there. This cell, this one cell undergo one mitotic division, one unequal mitotic division, unequal mitotic division to give one smaller cell, one bigger cell. Look at here. Here there is one more nucleus here and this is cell. You get my point? So here one smaller cell will form and one bigger cell will form. Unequal division. Okay. So this upper one is the bigger cell. This total one is the bigger one. Okay. And this smaller one, this smaller and this bigger cell will form. Smaller cell we call generative cell. Important, generative cell. The bigger cell we call vegetative cell. Vegetative cell, or it is also called tube cell. It is also called, tell me, tube cell. From this vegetative cell or tube cell, pollen tube will come out. Pollen tube will emerge. Keep in your mind. Now, how many cells are there in the pollen grain now? 2 cell stage. Yes or no? One is called the bigger cell, larger cell called the vegetative cell, and the smaller cell is known as the generative cell. So at this stage, exactly at this stage, from the anther, this is anther, just now only we have seen the microsporogenesis, right? From the anther, from microsporangium, this anther undergo dehiscence at 2 cell stage. At 2 cell stage, pollen grain will release to outside. Approximately 60% of angiosperms. Approximately 60% of angiosperm at 2 cell stage, pollen grain will be released to outside. The 2 cells are generative cell, vegetative cell. And the rest of 40% of pollen grains are released at 3 cell stage. What is the third cell? Look at here. Let me take here, after this pollen grain release, once pollen grain is dropped on the stigma, after pollen grain reach on the stigma, this generative cell undergo one more mitotic division. One more mitotic division. Okay, mitotic division to form 2 haploidal male gametes. These are called male gametes. Keep in your mind. So in the examination you will get a question. What is that? Which of the 2 cells in pollen grain will give male gametes eventually? In the examination, if this question come, which out of the 2 cells in pollen grain? Generative cell undergo mitotic division. After it reach on the pollen grain, it will give 2 haploidal male gametes. So how many cells now? 3. So rest of 40% of pollen grains will be released exactly at the 3-cell stage before the reaching of stigma, but 60% of the angiosperm which are released at the 2-cell stage pollen grain, once they drop on the stigma, once they reach on the stigma, it undergo what? One more mitotic division to form total 3 cells in the pollen grain. You get my point, student? Right, fine. So look at here. What happened once it reach on the pollen grain? Okay, once it reach on the stigma, from this germ pore, okay, here there will be a pressure, am I clear, on plasma membrane, and intine there will be a emerging of a tube-like structure outside. This tube-like structure is known as pollen tube. Pollen tube. I hope you get my point. So pollen tube is emerging from vegetative cell or tube cell. Clear, students? And few more important point about the pollen tube, pollen grains. Pollen grains are preserved. Pollen grains we can store at minus 190 degrees Celsius in nitrocellulose medium. Am I clear? For long of period of time it will not going to be destroyed because sporopollenin is there. Sporopollenin is a world resistant substance. We can store this pollen grain for long of period of time. Number 2, pollen grains also supplement for the food. If we can take that, because nutritive food material is there in the cytoplasm, so we can use as a nutritive supplementary. One more important thing keep in your mind, pollen grains are also may cause allergy, especially the Parthenium pollen grain. The Parthenium pollen grains are cause a disease, the allergen disease called hay fever. Am I clear? Hay fever. Moreover, the last point about the pollen grain, study about the pollen grain, I can tell you, palenology. Palenology is the term, a study about the pollen grain. So this is called the male gametophyte structure or pollen grain structure. Exine, intine, plasma membrane, generative, vegetative cell, and keep in your mind for the MCQ's question, which cell is going to give a tube cell, pollen tube, which cell is going to give a male gametes. So this is the complete structure of male gametophyte. So in the next session, in the next video, we will discuss few more important questions from first term chapters. Thank you.